All right, good morning, ladies and gentlemen. It's morning, at least here in the classroom, 7:07. We're going to talk about RNA and protein synthesis. I'm going to have to split this into two sections because it does take a little bit of time. So we'll start with RNA. What exactly is RNA? It sounds like it's DNA, so we're wondering what that could possibly stand for. RNA stands for ribonucleic acid, and if you remember what DNA stood for, it stood for deoxyribonucleic acid, and deoxyribose was the name of the sugar in the sugar phosphate backbone. Do you remember it went sugar, phosphate, sugar, phosphate, sugar, phosphate? Well, ribonucleic acid, instead of having deoxyribose as its sugar, has ribose as its sugar. Well, what is RNA? Let's find out. There are 4 bases in RNA, and so it has a lot of similarities to DNA, and yet there's a lot of things that are strangely different between RNA and DNA. RNA has cytosine, C, guanine, G, adenine, A, and uracil. Where did uracil come from? Uracil, they think, is a base that takes less energy to build, and so the RNA uses uracil instead of thymine. The cell is willing to expend a lot of energy to build DNA, so it spends a little more energy on the thymine, a little more stable molecule, and it saves a little bit of energy on the RNA. You don't know why yet? That's okay. RNA is also single-stranded. It only has one side. Look at that diagram. It's missing the second side, so it's not a double helix like the DNA is. So one more time, how does RNA differ from DNA? First of all, it's single-stranded. It has uracil in place of thymine, and it has ribose in place of deoxyribose. Take a look at the 2 diagrams and see if that makes sense. And let's move on and talk about the different kinds of RNA and how they're relevant. There are different kinds of RNA. The first one that we're going to learn about is called mRNA. mRNA stands for messenger RNA. The M is lowercase and then the RNA are capital. And messenger RNA is a copy of one side of the DNA, one of your genes, and it carries a message from the DNA out of the nucleus and out to the cytoplasm of the cell where there's a ribosome. And the ribosome reads the mRNA and then builds a protein. So the DNA stays safely in the nucleus where it's less likely to get damaged. And then the mRNA is a messenger, like a bike messenger or something, that travels out and delivers blueprints, let's say, for a building. And if it gets damaged, the blueprints get damaged, we can always make another copy of the DNA. The DNA has to stay safe in the nucleus. So the mRNA is a copy of it. The next kind of RNA is called transfer RNA. I often refer to transfer RNA as the trucks because tRNA also starts with the same letter as a truck, T-R-U-C-K. The trucks here are carrying the building supplies for making this amino acid, or in my analogy, I say it's building a building. Trucks carry amino acids for building a protein to the mRNA at the ribosome. At the very bottom of the tRNA, you should notice what's called the anticodon loop. It has the opposite letters that RNA has, the mRNA has, and so the mRNA tells which truck needs to pull up, and that'll start to make sense as we go. The last kind of RNA is called ribosomal, or rRNA. I will never call it rRNA again because it's awkward to say, rRNA. Those are the ribosomes, and that's what we're always going to call it, the ribosomes. It's RNA that's all kind of mushed up in a globular form. This is where proteins are made. In this artist's rendition, the kind of giant green blobs are the ribosomal RNA. The strip that's running through and then curling at the end, that's the mRNA. And then the circles, the spheres all coming together, that's the protein that's being built out of amino acids. All right. Transcription is a process that we're going to talk about, and actually we may as well talk about that right now. I'll talk about translation on the next one. Transcription is the process of actually making RNA. There's another process that we're going to talk about in a few minutes called translation, and that's building the protein. So transcription is just making the RNA. So this is what happens. You remember when DNA gets replicated, that the DNA untwists and it unzips. The 2 sides get separated from each other. Well, the same thing happens here, Only this time when it untwists and unzips, instead of DNA polymerases attaching to both sides and copying both sides, we have a new enzyme called RNA polymerase. RNA polymerase only attaches to one side of the DNA. They think the second side of the DNA molecule is just there for structure to make it a more stable molecule. So the mRNA or the RNA polymerase only copies one side of the DNA. And it lands at what is called the promoter site, an area on the DNA that is sort of like this is the beginning of a gene signal to the RNA polymerase. So it attaches there, and then it reads the DNA one letter at a time, just like DNA polymerase did, and it attaches one RNA nucleotide at a time. So remember that RNA has A, C, G, and U. So across from the DNA, if there was an A, you would have a U instead of a T when the mRNA is being made. That band, that red band in the diagram, is the mRNA being formed as the RNA polymerase is moving through. RNA polymerase continues to copy this one side until it reaches a termination signal on the DNA. A termination signal is a pattern of letters, a pattern of A's, T's, C's, and G's that tell the RNA polymerase, now we're done. And so then it lets go, and the mRNA is completed, and the mRNA breaks away from the DNA, leaves the nucleus, and heads to a ribosome. All right, that's where we're going to stop this set of notes. The next set of notes tells us when the mRNA leaves the nucleus, how it helps build a protein.